my name is Brian Ackley. I am an associate professor at KU in the Department of Molecular Biosciences. I teach for juniors, seniors, and graduate students. I started in college and as an engineering major, and I had to take a class on developmental psychology. And I can still remember sort of sitting on the floor reading a chapter where they introduced the idea of synapses. And then I was really I had to know more about them. And so pretty much everything that has led me from that point as a sophomore sitting on a floor in a lecture hall to where I am today was because of that sort of pivotal moment. So I teach a number of different classes, but my favorite to teach is my advanced neuro class. And that is a capstone class for seniors who are in the MCDB curriculum. And it's really focused on trying to understand the development, dynamics, and diseases of the nervous system. So we really try and integrate early, middle, and late sort of life effects on the nervous system. So I really hope that when students take that course, we can answer two questions. Number one, what are the sort of mechanics by which the nervous system forms and changes and adapts? But then really, the way that I set the class up is that we do a lecture and then we do a reading from the primary article, like primary literature. So most of the people that are taking that are juniors and seniors hoping to go on to graduate school, medical school, or something else. So I really want them to answer the question of like, how do we know things? Like, where does knowledge come from? When people go into the lab, how do they produce knowledge? And I try and sort of, contrast that with how things are presented in textbooks, where it's sort of like, somebody has taken the effort to go through all of that material and compress it into these sort of like nuggets of information. But when we're reading them in the primary literature, there are mistakes, there are things that haven't been corrected for. So I really want students to get a sense of how the neuroscience literature has evolved over time. And just as an example, one of the first things we do in class is we pull up the PubMed and we type in neuro as a stub. And we look at the fact that, there are like 70,000 articles that are just published in the last year. So I make the point is I can't teach them everything about the nervous system. So how can I empower them to use the literature to educate themselves about the things that are important to them? So I think that the great thing about KU biology, one is the professors that we have here are all, incredibly dedicated to both their research and teaching the students, but also the breadth of the professors that we have for the size of the department that we have. So we really go from, computational approaches that are very, very, focused on very small moments in time to organismal dynamics. And so I think having access to professors with that repertoire of skill set really sets KU biology apart from other places. Okay, so my lab has adapted a little bit since I've gotten here, but my long-term research interest has again been in sort of the fundamental mechanisms of how nervous systems form and how they change over time. And so to do that, we use C. elegans as a model system because they're transparent, so we can see things that happen inside of them. We can use fluorescent molecules. But I really kind of stumbled into it, which is, I think, how most geneticists get into their area of focus. And that was we found mutants that changed how the nervous system looked. And from there, we were able to sort of just, keep walking down the path. Since I have been at KU, though, because of the strength of immunology and genomics, we've actually added a question about how hosts and pathogens interact using, again, C. elegans. And in that case, we're taking C. elegans that are found from all over the world. We're sort of allowing evolution to do the genetic perturbations. And then we're asking how they are different with their responses between bacterial infections. And so as just an example of, again, going back to why KU is a great place, that wasn't something that I had ever envisioned working on when I even got here. And so, having colleagues that have helped me grow as a scientist has really been great. I do. I mentor students from many different levels. So right now, my lab has one student who's actually, well, I have a student who is a junior high student who comes into the lab occasionally, who's trying to get a sense of science. I have about 7 undergraduates, 5 graduate students, and 2 people who have completed their PhD and are working as postdocs. I think my mentorship style is really individual. So a lot of students are at different places in their career, but that overall, the things I try and focus on are empowering students to feel like they're learning by making mistakes successfully, right? And so, like, one of my favorite examples of that is one of my first graduate students, who I showed him how to do something on the microscope, scoring a phenotype. And then he came to me and said, I think I'm seeing something new, but I didn't know what I was looking for. So can you tell me if this is new? And it turns out no one had published any papers on that phenotype. And it's turned into a whole new area of research for us in the nervous system. So I really try and be supportive, and I really try and make sure that students know that it is okay to fail. It's expected. And that from those, if we can learn to fail successfully, then we're doing our job. So students in my lab learn how to rear and maintain C. elegans. They learn how to do genetics. So we do a lot of stuff with either loss of function or gain of function mutations. And in doing so, you have to understand sort of simple things about, like, how do you mix genetic backgrounds together? How do you isolate those that are appropriate? That might be phenotyping or genotyping using PCR sequencing. We do a lot of microscopy because these things are very small. They're about the size of a comma on a regular piece of paper if you're reading the book. So everything we do is either under a dissecting microscope, or we have a laser scanning confocal microscope where we get really close to things like synapses and axons. And then we also do some more physiological stuff. So right now we have a project where students are learning to tell whether or not neurodegenerative models have defects in learning. And that was, again, something that's been novel here. So a lot of it is working with C. elegans, learning to do the basics, plus hopefully something that is fun. Like the first time students look at GFP labeled axons inside living animal, like, whoa, that's so cool. I live for that moment. I really love sports. So I play basketball and I play soccer, and I serve on the Lawrence Adult Soccer League board. I love to cook. I really like trying new recipes and going out to eat in Lawrence. And I do trivia. So I'm a big pub quiz fan. Ooh, that's a great question. A favorite place to visit in Lawrence. I like to ride my bike around town on the Lawrence Loop, and my favorite part to ride along is the riverfront. And I'm excited about some of the possibilities that people have talked about the riverfront undergoing in the next couple of years. So I'll say that, and then a close second would be just downtown, Mass Street. I would say my favorite class as an undergrad was probably physical inorganic chemistry, which is weird because I was a chemistry major and I don't do physical inorganic chemistry, but it was a fascinating class. I am a really proud dad. I love my kids, and if anyone said you had to choose one thing in your life, I would rather be a dad than anything else.